In the previous lesson, we took a bit of a closer look at glycolysis, and now we're going to move on to the Krebs cycle. So to review, in glycolysis, glucose was partially oxidized, meaning it had some electrons removed, to form pyruvate. Glucose is a 6-carbon molecule, and pyruvate is a 3-carbon molecule, and so we made two 3-carbon pyruvates. We also got out 2 ATP by substrate level phosphorylation, and then the 2 NADH as a result of that oxidation. Now we're going to take the pyruvates through the Krebs cycle. Now, I'll mention that the Krebs cycle has a number of different names that you might come across if you're doing any reading or looking online. It's also called the citric acid cycle because citric acid is an important intermediate in the Krebs cycle, and it's also called the tricarboxylic acid cycle or the TCA cycle because citric acid is a molecule with 3 carboxyl groups on it. So just to be aware that these all mean the same thing. Now over here on the right, I have the symbols that I've been using to orient us to the various steps in cellular respiration, and this one was glycolysis, which we've already completed. This one now is the Krebs cycle, which we're going to look at in this lesson, and this one down here is the electron transport chain. Recall that glycolysis happened in the cytoplasm. If this is a mitochondrion, then glucose was split to 2 molecules of pyruvate out here in the cytoplasm, not actually in the mitochondrion at all. But now we're going to move into the mitochondrion, because the Krebs cycle occurs in the mitochondrial matrix. Remember that the matrix is this in here. It's like the cytoplasm of the mitochondrion. So pyruvate, which was made out in the cytoplasm, needs to be transported into the mitochondrial matrix for the Krebs cycle to occur. And the Krebs cycle is going to completely oxidize pyruvate to make carbon dioxide. So all of the carbons that started out in the glucose and that are now in the pyruvate are going to leave in carbon dioxide before the end of the Krebs cycle. And we're going to strip off electrons and put those on high energy electron carriers, NADH and FADH2. That's the oxidation. Now the first step in that is moving pyruvate into the mitochondrial matrix. That happens by a transporter, but then after that we have an enzyme catalyzed reaction called the link reaction. Called the link reaction because it is effectively linking glycolysis and the Krebs cycle. And a molecule called CoA, coenzyme A, comes in, and I won't draw you or show you the structure of CoA, and we oxidize pyruvate and get out NADH. So NAD plus comes in and strips off electrons, and we make NADH. Remember, this is a high energy electron carrier, and we also lose a carbon. So this is a 3-carbon molecule. We lose a carbon here off to carbon dioxide, and the product of this reaction is a 2-carbon molecule. Here's my 2 carbons attached to CoA. And again, we won't draw the structure of CoA. This is known as acetyl-CoA. Acetyl because it's got a 2-carbon group, and CoA for the coenzyme A part of it. This reaction, again, is called the link reaction because it links glycolysis in the Krebs cycle, but it's also known as pyruvate oxidation because we're stripping electrons off of the pyruvate and they're ending up here in NADH, and it's also called pyruvate decarboxylation because we are taking a carboxyl group off of pyruvate, and that carbon here is ending up in CO2. So we'll call it the link reaction, but you might see it called these other things as well. So now we've got 2 of the 3 carbons. We've got 2 of the 3 carbons from pyruvate over here, and we've lost 1. Acetyl-CoA acts as a shuttle to bring those 2 carbons into the Krebs cycle proper. And the molecule that accepts them into the cycle is called oxaloacetate. So oxaloacetate is a 4-carbon molecule, and then we get the 2 carbons in here from acetyl-CoA. So now we've got a 6-carbon molecule again. Now there are a lot of reactions in the Krebs cycle. Again, I'm not going to show you what they are, and I'm going to just show you what comes off of the cycle. Again, I'm not going to put these in really the correct order. I'm going to collapse them together. So one of the things that comes off of the Krebs cycle is NADH, and in fact we get 3 NADH for each acetyl-CoA that has come into the cycle. So 3 NADH. We also get out 2 CO2. So the 2 carbons that came in up here are both lost in carbon dioxide. So if you imagine we started with a 4-carbon molecule joining a 2-carbon molecule, making a 6-carbon molecule, now we've gotten rid of those 2 carbons again, and we're back to a 4-carbon molecule, and the process can repeat. Again, I'm not putting these in the correct places, I'm just showing you the different things that come off. Another thing that happens in one turn of the cycle is we get off 1 ATP, and we also get another high energy electron carrier that we haven't seen before but is like NADH. It's called FADH2, and we get one of those off. So again, for one turn of the cycle, we started with a 4-carbon molecule that accepted a 2-carbon molecule that gave us 6 carbons, but then we lost 2 carbons along the way as they both came off as carbon dioxide. So we could restart the cycle again with a 4-carbon molecule. But importantly, we got out some high energy electron carriers, NADH and FADH2, and we also got out an ATP by substrate level phosphorylation. Now the numbers that I'm showing you here happen in one turn of the cycle. That happens from 1 acetyl-CoA that was from 1 pyruvate. But remember, we got 2 pyruvates for every glucose. So all of these processes, including the link reaction, happen 1 time per pyruvate, but because there are 2 pyruvates per glucose, they're happening twice per glucose. So there are 2 turns of the Krebs cycle per glucose, and if we include the link reaction and the Krebs cycle together, then for example you can see that instead of just 1, 2, 3 carbons leaving, we've got a total of 6 carbons leaving, again because all of this is happening twice per glucose. And you can see then that all of the carbons have left, all the carbons from glucose have left in carbon dioxide by the end of the Krebs cycle. And you could do the same kind of accounting for the NADH and the FADH2, and for this ATP that's being formed by substrate level phosphorylation. So again, the Krebs cycle turns twice per glucose, once per pyruvate. If we do an accounting here with our overall equation for cellular respiration, then we had already in glycolysis split our glucose into pyruvate. Now we fully oxidized the pyruvate, and so there's no more pyruvate anymore. All the carbons have ended up over here in carbon dioxide. So now we've formed all the carbon dioxide that we're going to form in aerobic cellular respiration. It's all formed in the link reaction and Krebs cycle. We haven't used any oxygen yet, and we haven't made any water yet. Those are coming up later in the electron transport chain. In terms of the energy now, we have a bit in ATP. We have lots in NADH and FADH2 now. So there's no more carbon molecules, all the carbon's gone as carbon dioxide. The energy from the original glucose is mostly in these high energy electron carriers, NADH and FADH2, and we've made a little bit of ATP by substrate level phosphorylation here as well as in glycolysis. So now what we need to do is move to the electron transport chain so that we can see how the energy that is still stored in these high energy electron carriers is transferred to ATP.